over here. Actually, it looks like my headset's gonna die. Let me get the charging cable real quick. Okay, there we go. And switching views over here.
All right. Let me see. Can you hear me? Yeah.
Actually, it looks like my
Okay.
headset's gonna die. Let me get the charging cable real quick.
Sorry, having technical difficulties. There we go. I had the video pulled up and I can hear myself. Anyway, talking about the lymphatic system, we briefly mentioned that during the vasculature unit. Um, so they're a vast collection of cells and biochemicals that travel through lymphatic vessels. So the lymphatic system contains a network of vessels that assist in moving fluids from the tissues back into circulation. And as a result, they're closely associated with the cardiovascular system. Of course, the lymphatic system also has a role in immunity, so it helps defend the body against diseases. It transports, as we mentioned, that excess fluid away from the interstitial space, away from the tissue fluid, and returns it to the bloodstream. And we didn't mention, but there are lymphatic capillaries in the digestive system, which help move defense against disease and permanent immunity against future infections. And you're going to see that in the lymph nodes, we have B and T cells residing there. And they're basically monitoring the fluids that are picked up from the tissues, and they're looking for signs of infection. All right, so as far as lymphatic pathways go, lymph flow goes, it starts in the lymphatic capillaries. Of course, you saw how we have the capillaries here, okay, in the tissues, the arteriolar end, venular end, okay, we bring oxygenated blood to the tissues, dump off that oxygen as well as fluid, electrolytes, nutrients, and other small molecules. And then at the venular end, we're picking up some of the water and returning that to the bloodstream. But not all of the water gets picked up by the capillaries. The the capillaries. So the lymph capillaries down here pick up the remainder of the fluid and transport it through lymph vessels to lymph nodes, then to larger lymph vessels, and then finally into lymphatic trunks, ducts, and then we return it to the subclavian vein, uh, which dumps into the superior vena cava and then returns to the right atrium. Okay, and so that excess fluid is picked up in the tissues by the lymphatic capillaries, carried through the vessels, lymphatic vessels to the lymph nodes, and then from there through larger lymph vessels, ducts, etc., back to the subclavian vein and into the superior vena cava. Okay, returning that fluid back into the bloodstream. All right. So these are the main lymphatic capillaries. There's another type of lymphatic capillary called a lacteal. Lacteals, we mentioned on the last slide, they're found in the digestive system, specifically in the small intestine. And what they'll do is pick up fatty acids, pick up lipids, and transport them to the liver to get processed. Basically, help bring them into the bloodstream and the body. All right, so the lymphatic capillaries are microscopic closed-ended tubes, which you can see here, and they're interwoven with the capillary bed. Okay, you can see the capillary bed here, and then you can see these finger-like projections. Okay, those are the lymphatic capillaries. They're thin-walled, composed of simple squamous epithelium, just like the capillaries, the blood capillaries are. Okay. So tissue fluid flows into the lymphatic capillaries. That's also known as interstitial fluid. And the fluid, once it enters the lymphatic capillaries, is now known as lymph. And then these lymph capillaries merge into these larger lymphatic vessels down here. So lymphatic vessels are similar to veins, but they're even thinner than veins. They have the same three layers, though. There's an inner endothelial lining. The endothelium, again, is simple squamous epithelium. We have a smooth muscle middle layer, and then an outer connective tissue layer. Similar to veins, we will also see valves in the lymphatic vessels. And again, what the valves do is when fluid, in this case lymph, flows up past the valve, um, gravity's going to bring it right back down. But we have the valves which prevent the backflow of lymph. So every time lymph moves forward, it's held in place by a valve, okay, and not allowed to travel back down. Without the valves in the veins and without the valves in the lymphatic capillaries, our tissue fluid would just accumulate in our feet. Gravity would just bring it right back down. So the lymphatic trunks drain lymph from the lymphatic vessels, and they're named for the regions that they serve. You have the lumbar, the intestinal, the intercostal, that's between the ribs, bronchomediastinal, and that would be in this region. The mediastinum is a region which encloses the heart and the thymus up here. Okay. And in this region, you'll also see the bronchial tubes branching off. But we'll talk about that when we get to the respiratory unit. All right, so the lymphatic collecting ducts, they drain lymph from the trunks. So the trunks return fluid to the ducts. The ducts are large lymphatic vessels, okay, found just above the collarbone, and they're going to dump lymph into the subclavian veins, the right subclavian vein and the left subclavian vein. And those are going to dump, well, blood, which now contains fluid from the lymph, into the superior vena cava and then into the right atrium. Okay. So the thoracic duct, which is located on the left, and you can see it indicated here, is much larger than the right lymphatic duct. The right lymphatic duct over here drains into the right subclavian vein and is smaller. And we can see here why. The right lymphatic duct collects fluid from the right side of the head and the right arm and chest. Okay. So all of that dumps into the right lymphatic duct and then into the right subclavian vein. The thoracic duct, on the other hand, collects fluid from the left side of the head, the left arm, and left chest, but also the entire abdomen and both legs. So the thoracic duct is draining fluid from a much larger portion of the body and returning that fluid to the left subclavian vein. All right, so we've just talked about the flow of lymph. One word we should probably use here is afferent and efferent. We should point that out. So whenever you see the word afferent, think input, and efferent output, or afferent in, efferent out. So we saw those words when we were talking about the nervous system. You have afferent fibers, which bring sensory information to the central nervous system. And then you have efferent fibers, which carry motor information away from the central nervous system. In the lymphatic system, you're going to have these lymph nodes, and the vessels that carry fluid into the lymph node are the afferent lymphatic vessels, and the vessels carrying fluid away from the lymph node or out of it are the efferent lymphatic vessels. Okay? And then those continue on to the lymphatic trunks, which are regional, and they drain into the ducts, the right collecting duct and the thoracic duct, and then from there into the right or left subclavian vein. All right, so capillary blood pressure filters water and small molecules from the plasma. Basically, hydrostatic pressure inside of the capillaries, specifically at the arteriolar end where hydrostatic pressure is high, that's going to cause fluid and small molecules to filter out of the capillaries. So by small molecules, we're talking about anything small and lipid soluble. So oxygen and CO2, they diffuse quickly, although CO2 would be diffusing into the capillaries, oxygen out. Alcohol is also lipid soluble, so it would get through easily. Um, Fatty acids, cholesterol, things like that could get through easily. Polar molecules such as glucose and electrolytes, which are small, will be able to filter out into the tissues. And so the fluid in the tissues depends on the composition of the blood. So whatever concentration you have of electrolytes in the blood, it's going to be pretty much the same concentration in the tissue, pretty much. And then whatever concentration of glucose you have in the blood, you're going to have a similar concentration in the tissue, at least until the cells take it up. Um, yeah, so the only difference between tissue fluid or interstitial fluid, it's the same thing. Only difference between that and the blood plasma is that you're not going to see large plasma proteins. Okay, large plasma proteins, such as the albumins and the globulins, they're not going to make their way into the tissues. They're just too big. All right, what else? And then, of course, tissue fluid will not have red blood cells, or well, it won't have red blood cells. Sometimes white blood cells are able to get in. All right. Good. So tissue fluid again basically is blood plasma minus the plasma proteins. So filtration from the plasma normally exceeds reabsorption, as we said. More water flows out of the blood capillaries into the tissues than is picked up by the blood capillaries. So that leads to an accumulation of fluid in the tissues. All right, and so this excess fluid, as it builds up in the tissue, it gets it's under higher and higher hydrostatic pressure. In other words, as fluid accumulates in the tissues, the hydrostatic pressure in the tissue increases. All right, and so once the hydrostatic pressure in the tissue is high enough, the fluid will flow into the lymphatic capillaries, and the fluid, again, once it's in the lymphatic capillaries, is called lymph. Along with the fluid, we're going to see whatever's in the tissue fluid move into the capillary. So it's not just water. You're going to see electrolytes and other substances from the tissue fluid move into the lymphatic capillaries. Now, if excess fluid accumulates in the tissues more than can be picked up by the blood capillaries or the lymphatic capillaries, that results in a condition called edema. Edema is excess fluid in the tissues. That can lead to visible swelling and puffiness. All right, so lymph inside the lymphatic vessels has low hydrostatic pressure, meaning, in other words, lymph pressure is low. Similar to venous blood, good lymphatic flow requires help from a couple mechanisms. Just like veins, the lymphatic system relies on skeletal muscle contraction. When skeletal muscles contract, they compress the lymph vessels just like they compress the veins, and they push that tissue forward. Then we have the valves, which prevent backflow. Also, just like the veins, we're going to see changes in pressure in the thorax, moving fluid forward. As we breathe in, pressure increases, and as we breathe out, pressure decreases in the thorax. And that's going to propel lymph forward just like it propelled blood in the veins forward toward the heart. All right, so yeah, we mentioned the valves and the lymphatic vessels. Good. Lymphatic flow is going to be highest during exercise. Same with venous flow. All right, so here we see the tip of a lymphatic capillary, and one thing you're going to notice hopefully, is that these endothelial cells have little flaps, okay? Flap-like valves. Let's pretend they're capillaries down here, blood capillaries, okay? And at the arteriolar end, hydrostatic pressure is high, and so we filter water and other substances out of the blood into the tissue space, into the interstitial space. Some of that fluid gets picked up by the venular end of the capillaries and then moved up to the heart, right? Returned up to the heart through venous blood. But not all of that fluid gets picked up by the blood capillaries. Some of it remains in the interstitial space, in the tissue fluid. All right, and as fluid accumulates here, the pressure is going to increase. When the tissue fluid pressure becomes higher than the lymphatic pressure, fluid will flow into the lymphatic capillary through these valve-like veins. As the lymphatic capillaries and the blood capillaries pick up fluid, eventually pressure in the interstitial space drops. And when that happens, you might expect lymph to flow back out, but these flap-like valves close and prevent the fluid from flowing out. Okay? Okay. I think that's about all I wanted to say about that. Um, okay. So in addition to the lymph nodes, we have a couple lymphatic tissue types. So there's the mucosa-associated lymphatic tissue. This is unencapsulated tissue found in the digestive, respiratory, urinary, and reproductive tracts. And as the name might tell you, basically you'll find it associated with mucosa, where the mucosa are. In the tonsils and the appendix, we have a number of lymphatic nodules. And then we have Peyer's patches in the third and final, or the distal, part of the small intestine. Okay, these Peyer's patches are found near the end of the small intestine and the beginning of the large intestine, and their job is to catch bacteria that are trying to move from the large intestine into the small intestine. We want to keep bacteria in the large intestine. They help us out there. Uh, but bacteria in the small intestine would rob us. They would rob us of the chance to absorb our nutrients. So we want to have the first opportunity, first choice of nutrients as they move through the small intestine, and then the bacteria can have our leftovers when they get to the large intestine. All right, so then we have lymphatic organs, which have the encapsulated lymphatic tissue. They include the lymph nodes. We also see the thymus and the spleen are lymphatic organs. And so here is a view of an actual lymph node, and you can see the lymphatic vessels entering and exiting it. Over here, you can see the cartoon depiction of a lymph node. Here you have the afferent lymphatic vessels carrying fluid into the lymph node. And then down here you have the efferent lymphatic vessels carrying fluid out of the lymph node. So this fluid enters the lymph node and it's bringing with it, it's not just water, of course, it's not just nutrients, it's not just electrolytes. We're also going to see, tiny molecules that were just floating around in the tissue. Some of that is waste, some of that is remnants of dead cells, and some of that might be little pieces of bacteria or something like that. If we had an infection going on, bacteria, may die in the tissue or they may release a toxin into the tissue, and that gets picked up by the lymphatic flow and then brought to the um, lymph node. And in the lymph node, we're going to have B and T cells, which are sitting around monitoring the fluid that flows through here, monitoring the fluids coming from the tissues. And if they see antigens, which are things that elicit an immune response, if they see antigens from bacteria, fungi, etc., they'll respond to it. Some B cells and T cells attack bacteria actively and they'll go into effect. Then, of course, we have the memory B and T cells, which stick around for years so that we can maintain an immune response down the road if we see this infection again. The B cells, of course, produce antibodies, which can flow freely through the blood and attack tissues and mark them, not tissues, flow through the blood and mark pathogens for destruction or directly attack them. Then we have the T cells, which recognize those antibodies and destroy the invaders, destroy the pathogens. All right, so here we see the lymph node again. All right, so the locations of the lymph nodes, they're distributed throughout the body, but not evenly. We have a high concentration in the inguinal region, the axillary region around the breast as well, along the neck and head. Usually, when people think of lymph nodes, they think of in the neck. Someone asks you, "Are your lymph nodes swollen?" and someone immediately reaches for their neck, trying to feel it. But you'll also find them in the supratrochlear region, here above the elbow. Okay, you'll also find abdominal and pelvic lymph nodes. All right. So, two functions of lymph nodes. They're going to filter potentially harmful particles from the lymph. That could be toxins or wastes or just cellular debris. And they're also going to perform immune surveillance, as we talked about. They monitor the body fluids for signs of pathogens. Along with the red bone marrow, which is where lymphocytes are initially produced, lymph nodes aid in the maturation and production of lymphocytes. So the lymphocytes we mentioned, they attack various pathogens. The T cells directly attack them. The B lymphocytes produce antibodies against them. And then we also have in the lymph nodes macrophages. Now, macrophages actually come from the monocytes, which are part of the myeloid stem cell line. So this is way back in the blood chapter, but different cell line, also starting in the bone marrow, though. These macrophages, they surround, they phagocytize, in other words, they eat foreign substances, cellular debris, damaged cells, and they'll also eat bacteria and fungi and try to destroy them. So the thymus is this little organ right here, located superior to the heart. Okay, here's the thymus. And it's divided into lobules. It's located in the mediastinum, which includes, again, the heart, the thymus, here, heart and thymus. The lobules contain lymphocytes that come from progenitor cells in the red bone marrow. Basically, the thymus is the site of maturation for T cells. B and T cells get their start in the red bone marrow. B cells will mature in the bone marrow, and T cells will mature in the thymus. Then you may remember from the endocrine unit that the thymus also produces thymosins, which are a local hormone that stimulates T cells to mature into T cells. So the thymus is going to be large in infancy. When you're born, the thymus is enormous, and it's going to shrink at puberty. In the adult, it's pretty small, okay? And then in the elderly, it's going to be replaced over time with adipose and connective tissue. Now, we've talked before about the spleen and how it serves as a blood filter, which breaks down old red blood cells and then passes the waste onto the liver to get processed. The spleen is also the largest lymphatic organ. And it's located in the upper left part of the abdominal cavity. It's kind of supero-posterior to the left kidney. and it looks like a large lymph node under the microscope. It contains sinuses that are filled with blood and two kinds of pulp. We have the white pulp, which is made up of lymphocytes, and you can see that here. Looks pink. Now remember, when we look at these microscope slides, almost everything in the body is more or less clear. When we get a thin slice of it, it's going to look clear. It looks pink or purple here because we've applied dyes and stains to the tissue so that we can see it better. But anyway, on the slide, the white pulp is kind of pinkish, and then the red pulp out here, that's going to be made up of, yes, also lymphocytes, but red blood cells and macrophages as well. All right. So the spleen, it filters blood just like the lymph nodes filter lymph. All right, and good. So the lymphatic system, we said it has a role in immunity. It defends the body against infection and pathogens. So pathogens, you probably know, these are disease-causing agents, things like bacteria, viruses, and then you can have complex microorganisms like protozoa. Protozoa, you may not have had a biology class yet, like a college level biology class yet. But protozoa are in the same kingdom as fungi, plants, and animals. Eukarya. Um, Actually, that's not a kingdom; it's domain. But anyway, we're in the same super family. All right. We have similar cell types, in other words. Bacteria and archaea, which is potentially another pathogen, have much more simple cell types, and then viruses technically aren't even alive. But so that was a little bit of a side rant, I guess. Back to the topic. Pathogens include bacteria, viruses. You can have protozoa, protists like euglena, and then fungi. Not just the spores. Kind of odd it mentions spores, but fungi can infect people. Often it's yeast-like fungi. Now, that's not baker's yeast. That's not brewer's yeast. That's not the kind of yeast we have in the kitchen. It's a different kind of yeast. All right, so the presence and multiplication of a pathogen in the body can cause infection. Basically, these pathogens divide and they rob the nutrients of tissues. Sometimes they directly attack and destroy cells in the body, and all of this elicits an immune response that results in fever, which is part of the innate or non-specific defenses. Uh, and then you'll also see neutrophils and other white blood cells infiltrating the tissue and attacking the pathogen, depending on what type it is. And then we have the adaptive or specific defenses, which are known as immunity. These are more specific, they're more precise, and they target specific antigens. So here we're looking at the B and T cells mainly. All right, so this is carried out by the lymphocytes, the B and T lymphocytes. They recognize certain foreign molecules. As we mentioned, the B cells produce antibodies against them, and the T cells directly attack them. So here we're going to talk about immunity and a couple types of innate or non-specific defenses. Let's back up a second. Why do we call it innate or non-specific defenses? Well, innate means that it's built in. Non-specific means that this defense is not targeted for a specific bacterium. If E. coli, no, that's not a good example. If Staph aureus tries to get into the body, it's going to run into one of our biggest non-specific defenses, skin. Skin isn't there to keep Staph aureus out. Skin's there to keep all bacteria out. So skin is non-specific because it's not targeted against a specific bacteria. It keeps all bacteria out. All right. So the first one we're going to talk about though is species resistance. So in my biology class last week, I was explaining to the students about coronavirus and why is it kind of scary. First, it's slightly more fatal than the flu. A lot of people brush that off like the flu's nothing, but flu killed 8,000 people last year. That's not nothing. But another reason the coronavirus is scary right now is because there's a list of diseases that affect humans. And we don't want to add a new one to that list. We don't want to add new diseases to that list. So the goal right now is to stamp it out completely before it's so widespread in the population that it's going to be with us forever. Um, all right, so back to species resistance. There's certain diseases humans can get, and there's certain diseases that humans can't get. Like you're never going to catch feline immunodeficiency virus. Yeah, cats have a immunodeficiency virus. They have their own version of feline AIDS, and I mean you're never going to catch that. Um, well, I say never. It's hard to say never in biology, but you're not going to catch that. Same as a cat is never going to catch human immunodeficiency virus. A cat is never going to catch HIV. Dogs have their own form of hepatitis. There's canine hepatitis, and you're not going to catch canine hepatitis, and dogs aren't going to catch human hepatitis. So, where does this come from? Basically, the bacteria or viruses or fungi, whatever pathogen is responsible for this disease, um, it lives well in a certain host. So, some diseases love to live at 98.6 degrees, which is human body temperature. And once they get to that body temperature, once they get to that temperature inside our bodies, then they go to work attacking. But dogs have a different body temperature, and if a human disease entered a dog and it was waiting for that signal to reach that signal that it was in a dog, I'm sorry, a human. If it was waiting for the temperature to reach 98.6 degrees, that may never happen, and so it may never get activated. There are other things too. These pathogens look for cell markers so that they know what cells to invade. And those markers for a human disease are going to be totally absent in a dog. And so the cell, let's say bacterium, uh, doesn't know it's in a host because it's in a dog. It doesn't know to attack the tissues because it's in a dog. It's not seeing any of its familiar markers. All right. So I'm sorry if that little rant was confusing. It's a complicated topic, but basically, certain diseases only affect certain animals. It is possible for a disease to jump from one animal to another, that is, from one species to another, and that's what happened with swine flu, avian flu, and then actually also coronavirus and Ebola. It's believed that both of them actually came from bats originally. The coronavirus and Ebola, that is. Um, but these events are exceedingly rare, and they require they just happen when there are random mutations that allow it to happen, and you can prevent that by staying away from livestock and wild animals. Anyway, back to this. So we also have mechanical barriers. I mentioned the skin a little bit. The skin and mucous membranes, they form mechanical barriers, physical barriers that keep skin out. They're non-specific because they keep out all pathogens, or ideally, right? So they're considered the first line of defense against all other. I'm sorry, all other non-specific defenses are part of the second line of defense. They're the first line of defense against all bacterium and fungi and viruses. It's a physical barrier that keeps them out. The epidermis, of course, is made up of multiple layers, many layers of cells, and as they slough off, or basically fall off, they remove the bacteria that was sitting there on the surface of the skin. We also have ciliated epithelia in the respiratory tract, and those cilia, when they beat, when they move, they move mucus up the respiratory tract, and then that mucus gets moved with the bacteria and other things we breathed in, gets moved over to the esophagus. Then we swallow that mucus. It gets taken down to the stomach. We recycle the mucus and we will dissolve the bacteria in stomach acid. Hairs in the nose help trap pathogens, help keep them from reaching the respiratory tract. And then tears, saliva, and urine will all wash away microorganisms. Saliva actually contains some compounds which are antibacterial. Um, Obviously, you still need to brush your teeth. Bacteria have evolved to live well in our mouths, but anyway, there are also chemical barriers. So we have enzymes, which are in the body fluids, and they provide a chemical barrier by attacking the pathogen. So we have pepsin in the gastric juice, which dissolves proteins, and so it'll dissolve proteins in the wall of, say, a bacterium that entered the digestive tract. And we have lysozyme inside of lysosomes and inside of tears, which will destroy microorganisms. We have interferons, which block viral replication. They can also act against tumors and stimulate phagocytosis, which is again that eating by the macrophage. Defensins are peptides or short proteins that are produced by neutrophils and other granulocytes. They cripple the microbes by punching holes in their cell membranes. Collectins are proteins that protect against many bacteria, yeast, and some viruses. And then finally, complement proteins are a group of proteins found in the plasma and other fluids that stimulate inflammation. They attract phagocytes and they enhance phagocytosis. They can also come together, and it's pretty cool, also punch a hole in the wall of bacteria. And when you punch a hole in the wall of a cell, pretty much that's the end of the cell. All right. We also have natural killer cells. There's small populations of lymphocytes that are different from B and T cells. They defend against viruses and cancer cells by secreting cytolytic substances. Cyto means cell, lytic means splitting. And so these cells basically these substances split other cells. They're called perforins, the cytolytic substances. Perfor, short for perforate, right? So just remember punching holes in cell membranes again. So the natural killer cells also enhance inflammation, and the inflammatory response produces local redness, swelling, heat, and pain. It actually increases blood flow to the area. It causes vasodilation of the local capillaries, and as blood flow to the area increases, it's going to look more red, right? Uh, all right, so you'll also see swelling. If we increase blood flow, of course, we'll see fluid accumulate in the tissues. Then we see heat and pain associated with inflammation. All right, inflammation can wall off the infection site, and if you wall off the infection site, you stop it from spreading. Phagocytosis, we mentioned, is cellular eating, and it removes foreign particles from the lymph. Basically, macrophages eat cellular debris, they eat garbage that's floating around in the lymph. All right. Good. The most active phagocytic cells are the neutrophils and the monocytes. Remember, the monocytes, they travel through the blood, and when they reach a tissue and then move into the tissue, they mature into the macrophage. Okay. All right. So yeah, macrophages we mentioned. Mononuclear phagocytic or reticuloendothelial system. It consists of monocytes and macrophages that are distributed throughout the body. Monocytes in the bloodstream, macrophages in the tissue. Fever. So a fever is produced when a viral or bacterial infection stimulates lymphocytes to divide and increase in number. And then they produce cells that secrete a substance called interleukin 1. And interleukin 1 changes the thermoregulatory set point. In other words, our thermoregulatory set point, the place where the temperature we will maintain in order to maintain homeostasis, is around 98.6 degrees. Uh, and so interleukin 1 is going to raise that set point to 99. The body will attempt to maintain a temperature of, say, 99 or 100 or whatever, depending on the amount of interleukin 1 floating around. Interleukin 1 is also called an endogenous pyrogen. So, pyrogen, fire maker, endogenous from within, which elevates the body temperature, and elevated body temperatures inhibit microbial growth. Okay. They cause the liver and the spleen to take up iron. Why? Because new bacteria in your body need to divide and grow and multiply. And in order to do that, they need iron and other nutrients. And so if we take up all the iron out of the blood, that slows the rate at which bacteria can grow. All right. Good, good. And then the high body temperature also activates the phagocytes. Okay, so you can see here the divisions of the innate defenses. A pathogen enters the body and it encounters the first line of defense. We've got mechanical barriers such as the skin and the mucous membranes. Now let's say there's been a break in the skin or the mucous membrane. Now the pathogen can get into the body. We slip past the first line of defense. Well, now you're going to have to run into the second and third line of defense. The second line of defense, you've got everything we were just talking about, the chemical barriers, the natural killer cells, inflammation, phagocytosis, and fever. And then lastly, you got to run into the third line of defense, the cellular and humoral immune responses. We'll talk about those in a second. So the receptors on the surface of lymphocytes enable them to recognize non-self antigens, basically antigens on the surface of cells that are not human, not mine, right? So antigens can be proteins, polysaccharides, glycoproteins, glycolipids, basically anything capable of invoking or creating an immune response. And so the most effective antigens are large, complex molecules because they're easy to recognize. Haptens are small molecules that are not antigenic by themselves, but they can combine with a large molecule in the body and then evoke an immune response. Whoa. All right, so the lymphocytes are produced throughout life, starting with fetal development. The red bone marrow releases immature, unspecialized lymphocytes into the blood. Half of them settle in the thymus, which are then specialized and released. The other half of the cells differentiate in the red bone marrow and then are released. And remember, it's the T cells which specialize or differentiate in the thymus. And then the B cells, they will mature in the bone marrow. Now, T stands for thymus-derived lymphocytes, and then B stands for bursa of Fabricius in chickens. So just it's okay to remember B is for bone marrow. That's fine. The bursa of Fabricius is where the B cells were first discovered. That's why they get that name. All right. All right, so you can see that diagram here. We have the red bone marrow in the head of the femur here, and in the metaphysis and diaphysis. Excuse me. Sorry. So that's where progenitor early lymphocytes come from. They move through the bloodstream. Half of them will set up shop in the thymus and mature into T cells. The other half stays in the bone marrow and becomes B cells, then enters the bloodstream and travels to the lymph nodes. The mature T cells also travel to the lymph nodes, and in the lymph nodes they sit and they wait and they monitor tissue fluids. They monitor lymph. All right, so a lymphocyte has to be activated before it can respond to an antigen. Basically, it needs to see the antigen itself. The T cell activation requires antigenic fragments. That's fragments of the antigen, pieces of it, to be attached to an antigen presenting cell. An antigen presenting cell could be a macrophage, a B cell, or any number of other cell types that basically their job is to show antigens to T cells. They bring them to T cells and say, hey, look what I found, go investigate. The antigen presenting cell phagocytizes the antigen, basically engulfs it, digests it, and then sticks the antigen out on the surface of its own membrane. Okay. Now we can recognize that this is. So how do I phrase this? So if you eat a bacterium and then you put part of its cell wall out on your own plasma membrane, won't you look like a bacterium to the immune system? Well, not if that piece is attached to one of these proteins, the MHC or major histocompatibility complex, or the HLA, human leukocyte antigens. These are proteins on the surface of our cell, and if they're found with the bacterial antigen on the surface of the cell, then the T cells know when they see both of those that this is not a bacterium I'm seeing, it's a piece of a bacterium that's being shown to me. All right, so when the specific T cell encounters an antigen presenting cell, which displays the antigenic fragments, it can now recognize that antigen as foreign. So basically, if you show me a piece of Staph aureus, along with the MHC or HLA proteins on your cell surface, I've seen this piece of Staph aureus, and now the next time I see Staph aureus in the wild, next time I see anything that looks like this, I'll know that's a bacteria and I should destroy it. So there's a couple types of specialized T cells. You got the helper T cells, and they help activate other cells, and they help B cells produce antibodies. You have the cytotoxic T cells, cytocell toxic as well, toxic. So they attack virally infected or cancerous cells. Basically, some of your cells can send off a flare that says, destroy me. Like, I don't know what's wrong, but I'm going through something and I need you to destroy me. The body needs you to destroy me. So, for those cells which are cancerous or infected by viruses, cytotoxic T cells come along and they can recognize that and they will destroy the cell. Then finally, you've got memory T cells, and they provide future immune protection. How? Well, they remember the antigen, and then they stick around so that in the future they can respond if they see it again. All right, so B cells can be activated when an antigen fits into one of the receptors on their cell surface. Further B cell activation requires cytokines from the T cells. So once proper cytokines are released from helper T cells, the B cells respond by dividing and multiplying, enlarging clones of themselves. So some new B cells become memory B cells, which again stick around to recognize this threat again in the future. Other new B cells will differentiate into plasma cells, and plasma cells are the ones that release tons of antibodies into the bloodstream when we see an antigen. Okay. Since antibodies are carried by the blood, which is a body fluid, to the infection site, this response is called the humoral cellular response or the antibody-mediated immune response. Okay, so it's mediated by the humors, specifically blood. So we call it the humoral immune response. It's also mediated by antibodies, so we could say the antibody-mediated immune response. All right, so here we see a phagocyte eating a bacterium. Excuse me again, sorry. So we see a phagocyte eating a bacterium. It brings that into a lysosome or something similar, dissolves the bacterium. Now we've got these little pieces of bacteria inside of this organelle. We can then display that on the surface of the macrophage. So we do that. We display it on the surface of the macrophage. And then we're going to have T cells that interact with the macrophage. The cytotoxic T cell touches it, can recognize the antigen, and then it goes out and it attacks cells that are sending off signals that something's just not right. All right, so down here we've got the helper T cell, which has been activated because it now recognizes an antigen. And it's going to go off and it'll activate other cytotoxic T cells, or it'll activate B cells. All right. Now, after the infection's gone, the cytotoxic T cell can divide into another cytotoxic T cell or a memory T cell. And the memory T cells, again, they stick around and they look for infection in the future and can respond to it. Cytotoxic T cells are going to go right back out to the front lines and attack bacteria or fungi or viruses. Over here on this side, the B cell has to wait for the antigen to come to it. Basically, the bacterium comes to it, binds to a receptor on its surface, binds to an antibody on its surface, and it can now recognize the antigen, or in other words, the bacterium. So they're going to divide, and they're now activated. Okay, and when they're activated, they're going to divide and form either a memory B cell or a plasma cell. And the plasma cells produce tons of antibody and release them into the bloodstream to go attack more bacteria. All right. So antibodies, okay, these are proteins. They're Y-shaped molecules. And there's going to be, excuse me, a heavy chain and a light chain. The antigen binding sites are up here. Okay, so this is the region that binds to antigens, and which antigen it binds depends on the shape. Okay, and so you can alter the sequence of amino acids in a protein and change its shape, and the shape of this protein decides whether or not it can bind to a certain antigen. So for this one in the cartoon, the antigen it binds to would look something like a bell, kind of a bell shape here, right? All right. These proteins are globular proteins, also known as immunoglobulins. So they're part of the globulin family that you find in blood plasma. Um, Good. Each antibody is unique for an antigen, right? So if I'm releasing into my bloodstream antibodies that attack, let's say, the plague bacterium, if I later get an infection by Staph aureus, those plague antibodies won't do anything to Staph aureus because these are specific responses. Antibodies against plague will not do anything against Staph aureus because the antibodies against plague are specific for plague, not specific for Staph aureus, won't even recognize Staph aureus. You're going to need other B cells and other T cells which haven't been trained yet against a specific bacterium to recognize that bacterium and then initiate a response. So there are five major types of antibodies. You've got the IgG, IgA, IgM, IgD, IgE in that order. And if you need an acronym, the one I use in my head is GAMED, G-A-M-E-D. That helps you remember the order. Roughly, I mean, E and D you can reverse here. They're both less than 1% of all total antibodies. The most common type, IgG, makes up about 80% of the antibodies, and so it's the main immunoglobulin, the main antibody. The IgG antibodies, they circulate through the blood, they attack and they bind to and they mark for destruction all of the uh, pathogens that they're that they're specific for. So whatever pathogen they're specific for. All right. The M antibodies, so they react with antigens, but they're found on the surface of cells. Um, yeah, the D antigens activate B lymphocytes. So we don't need to remember all of these. The main one's G. The main circulating immunoglobulin is IgG. That would be good to know. So the antibodies, they react in a couple different ways. They can directly attack the antigen. All right. They can activate complement proteins and tell them to attack. And then they can stimulate local changes in the tissues. They can initiate inflammation, and they help prevent the spread of infection. All right. So immunotherapy makes the use of the immune system, to fight disease, both humoral immune responses by antibodies and cellular immune responses by cytokines. Cytokines are molecules released by cells as part of the inflammatory response. So you have monoclonal antibodies, which are a type of antibody. They're specific for a very specific piece of a bacterium. So, like the surface of a bacterium is not uniform. There are different regions, different pieces sticking out of it, and each antibody is specific for a different part of the outside of say, bacterial cell. Monoclonal antibodies are one type of antibody specific against one piece of a specific bacterium. Monoclonal antibodies are good because we can be very certain about what we're attacking when we use them. And we actually use them in a lot of modern medicines. There are a lot of new medicines, big area for pharmaceuticals coming out now, are what are called biologics, and many of them are monoclonal antibodies that are specific for something, right? And so you may hear about certain drugs like adalimumab, secukinumab, infliximab. Humira is the big one right now. That's adalimumab, and what they do is they attack inflammatory molecules. And so for people who have inflammatory conditions, chronic inflammatory conditions like psoriasis, you can actually give them a monoclonal antibody against uh, one of the inflammatory molecules. And if you do that, then you knock out inflammation. So that's one of the new drug types we're using. You can recognize them by their name. Anytime they end in MAB, that stands for monoclonal antibody, like adalimumab, secukinumab, infliximab. Anytime they end in MAB, that's a monoclonal antibody. We can also administer cytokines, or as I just mentioned, knock them out. They can be produced in large quantities due to recombinant DNA and monoclonal antibody techniques. We can use cytokines to treat leukemia, multiple sclerosis, hepatitis, genital warts, melanoma, kidney cancer. So here's a good chart. On the left, we see the primary immune response. So here we have our first exposure right around day 0, and then uh, we are measuring the plasma antibody concentration. That is the concentration of antibodies in the plasma over time. And you can see that it peaks after about 14, 15 days, something like that, right here. Okay. Um, so what does that mean? The first time you are exposed to a bacterium, let's say Staph aureus, the first time you're exposed to a bacterium, it's going to take up here about 14 days to reach the maximum response. Okay, so you could be sick for 14 days. You could have an infection in your thumb, an infected hangnail for 14 days. All right. That's the first exposure. Second exposure, I'm exposed to Staph aureus again. I've got another little hangnail, little staph gets in there. And look how fast the response is. It's within 5 days it reaches peak. And look how much larger the response is on my second exposure, right? Easily two, maybe three times the number of antibodies are now circulating in my blood. And with all these antibodies circulating in the blood, we're going to knock out that infection fast. In fact, we may never experience the symptoms of an infection because we're going to knock it out so fast. That bacterium can't take root. We're going to wipe it out within a day. All right. So why is this important? Because this illustrates how immunizations work, right? Why it's important to get vaccinated. It's right here. You can see it for yourself. What we're doing when we vaccinate children or even adults against a disease is we're taking a little piece of the bacterium or fungus or virus. Usually it's dead. You know, each vaccine's made in different ways. They make it as effective as they possibly can. Sometimes it has to be live virus or whatever, but it would be a small weakened version of the virus if it were alive, right? Normally, we for most immunizations, we just straight up kill the virus or bacterium or fungus, and then we will just give you pieces of it in an injection. And so you get these pieces of it, and the B and T lymphocytes recognize these pieces, recognize these antigens as foreign. And when we do that, we are going to activate T cells, of course, but we're also going to activate B cells and train them to produce antibodies against it. Now, training B cells to produce antibodies takes a long time, and so you can see here on this chart, reaching peak response takes 14 days. Getting all of our B cells up and going takes about 14 days. It's a long time when you're experiencing an infection in any way. In the case of a injection, in the case of an immunization, we just give you a small amount, not enough to actually cause an infection. If you were getting something live, and most of the time you aren't. Anyway, uh, but later, let's say in the real world, let's say I get an injection against tetanus. Let's say this is an injection against tetanus. Later in the real world, I step on a nail and tetanus gets introduced into my foot, passes right through the skin, gets introduced into the blood. My response is going to have B cells, memory B cells, rapidly producing antibodies against tetanus. Those B cells are going to rapidly produce antibodies against tetanus, and it's going to be wiped out before an infection can even get started. Okay? So that's the importance of immunization, and it also highlights the importance of those memory B cells. With the primary response, we develop not just cytotoxic T cells, helper T cells, all those. We also produce the plasma cells that produce the antibody and memory B cells that stick around and they remember the antigen so that the next time you see it, we can get ramped up really quickly. All right, so 4 types of immunity here. These can be kind of confusing, but they're actually not that hard to get. Let's say that immunity can be either naturally or artificially acquired, and it can be passive or active. Now, what does that mean? Naturally acquired immunity, basically, the name tells you, it's obtained through a natural process. When you're a little kid, you go out and you play in the dirt. You're going to be exposed to bacteria. That's naturally. When you acquire an immunity to the bacteria in the dirt, that's a naturally acquired immunity. When you're a kid, you get chicken pox. That's a bad example because chicken pox is with you for life. Um, when you're a kid, you get certain diseases, and then you're immune to them for the rest of your life. That's a naturally acquired immunity. Artificially acquired immunity would be through an injection. You get the immunity through artificial means instead of a natural process, and in this case, that would be something like an injection. Passive immunity okay, is where you receive antibodies. You don't make any of your own, you just receive them from another source. So we can give a person biologic drugs which contain antibodies. We can give someone an infusion of antibodies. Or, as a little baby, you drank breast milk, which contained antibodies from your mother. And then the immunity can also be active in that your own B and T cells are producing antibodies against the pathogen or the antigen. Okay? So you can have naturally acquired passive immunity. You can have naturally acquired active immunity. Naturally acquired passive immunity would be like mother's milk. You are receiving it from a natural source, mother's milk, and it's passive because you're receiving her antibodies, you're not making your own. Naturally acquired active immunity would be like we said earlier, playing in the dirt. We are exposed to bacteria, and then our B and T cells recognize the bacteria and develop their own antibodies against them. Now you can have artificially acquired passive and active immunity as well. Artificially acquired passive immunity would be where we're again giving someone an injection of antibodies. We're giving them an infusion of antibodies to treat an infection. And you can have artificially acquired active immunity, and that would be like an immunization. That would be a shot, right? Where we are giving someone a little piece of the bacterium, and then they develop their own B and T cells that will fight it off in the future. All right, let's see. Don't really want to get into hypersensitivity. Just real quick about autoimmunity. This is where the body recognizes its own cells as foreign for some reason. A lot of people now, I don't know how much research there is to back this up, but I've heard a lot of scientists say that now that we live in such a clean world and we don't face as much bacteria and infection as we used to, our immune systems are bored and sometimes turn against our own cells. Again, I don't know how much research there is behind that, but I've heard a number of scientists say it. And then now here I am saying it, spreading it. Anyway, so here, yeah, again, we produce antibodies against our own self. There are a number of autoimmune conditions where for some reason the body has recognized self as foreign. So a couple examples we've talked about are Graves' disease. We've talked about Graves' disease in the endocrine unit. There's myasthenia gravis we talked about in the muscular unit, muscles. Um, yeah, a lot of interesting conditions here. There's a list here. Um, you can look over if you like. So lifespan changes, just to finish up real quick. Here the immune system function is going to decline early in life when the thymus gland shrinks. It'll only be about 25% as powerful as it was. There's a higher risk of infection and cancer. All right, as we see a reduction in the power of the immune system, T cells, their numbers will decrease slightly, and then B cell numbers don't change, but their activity declines. They're less active as we age. The antibody responses to antigens become slower, meaning it takes longer for us to ramp up antibody protection, antibody production when we see a pathogen. IgA and IgG antibodies will increase, but IgM and IgE antibodies will decrease. And then the elderly may not be candidates for certain medical treatments that suppress immunity because of their weakened immune system. And treatments that suppress immunity include chemotherapy. All right. So for HIV and AIDS, HIV is the human immunodeficiency virus, and this is a virus that breaks down the immune system's function. It can stay silent for a number of years and then progress to AIDS, which is acquired immune deficiency syndrome. So HIV, just to be clear, is the virus. AIDS is the syndrome. What is a syndrome? Well, it's a collection of symptoms. So the condition is AIDS. The virus that causes it is HIV. And HIV, the virus, attacks macrophages and then helper T cells. CD4 and CD8 T cells. And when the helper T cell numbers decline, the B cells cannot produce antibodies due to the lack of activation by cytokines from the helper T cells. So they attack the T cells, and in doing so, prevent the activation of B cells. Later, HIV variants will affect cytotoxic T cells as well. And a person dies from the loss of the immune system, not the virus itself, but the loss of the immune system. Um, Also, some cancers become more prevalent because, again, the immune system has some role in suppressing cancer and tumor growth. So the modes of transmission are, of course, sexual contact, contaminated needles, birth or milk from the infected mother. When we say birth, we mean contact with the vaginal fluid of the mother through the birth canal. It can get in the eyes. And then receiving infected blood or tissues from a donor. All right. So we went a little longer than usual today, but that's it. If you guys have any questions, please feel free to email me. Nobody had any questions for the entire lecture today. All right. I'll send you an email out a little bit later about the exam next week as well. We'll get the study guide out. All right. Have a good day.